pleasure to be here. What I'm going to talk about today is follows on a study we recently, just 2 weeks ago, published in a journal called Cancer Discovery, that I think has garnered a bit of excitement. Certainly, we're very excited about it, but I think it also has a lot of potential for the way we treat cancer patients. As you'll see, it also builds on a bunch of research in this area and in our lab in particular over the past half decade or more. The overarching, if there's only one slide that you pay attention to for this talk, it would probably be this one because it gets the overarching idea out there. Why is it that we don't yet have a Siri or a Chat GPT that we can, or an Alexa or whichever you want? Why can't we talk to these things and instead of asking, for a restaurant recommendation or for directions to your doctor's appointment, why can't we ask it direct clinical questions about patient care down to the molecular level? So for instance, dear AI, my tumor or my patient's tumor has genetic mutations in PTEN, DAM1, and a whole constellation of genes, typically, depending on the cancer type. Those have been returned by what's becoming standard practice to sequence the cancer, if not the whole genome, at least large panels of genes. Insurance is paying for that at the moment for most or many tumor types, at least. But interpreting this profile ends up being a huge challenge. So wouldn't it be great if we could simply ask Siri what treatment is best given this molecular makeup? And then, that's the clinical version of the kind of AI you'd like to be able to chat with. The pharmaceutical version that you would use in a Pfizer, for instance, might be, what treatments are needed, and should we be looking at any new targets given this molecular profile of this person's disease. So this is essentially the AI version of precision or predictive oncology, which is really just the cancer version of precision medicine when you hear talk about precision medicine. I've bulleted here 3 major high-level aspects that you need to capture in such a precision medicine system. And then, in fact, everyone is interested in capturing it. Most people, I should say, in building precision medicine systems. The first one is obviously the system that you build has to be accurate. So given the patient record and molecular, including both clinical and molecular data, shown over here at left, you need to be able to make accurate predictions. So in the predictive challenge I'll talk about most today, predicting whether or not a patient will respond to a drug, then you would also feed the candidate drug, for instance, to the system, so it can render an opinion about whether that's a good idea. And first bullet here is you want to have a system that can accurately diagnose and recognize the patterns in the patient to make a reliable prediction. And in fact, there's been a lot of recent advances in cancer genetics recently and in AI for how to do that. What I'm going to talk about today is focus. I'll show accurate systems, or arguably accurate systems, but there is an alternate focus, which is related to the second bullet and to a degree the third bullet. Mechanistic interpretability and transferability. So what does this mean? Well, this means that it's not good enough, going back to this, talking to this Siri of the cell here, it's not good enough for Siri to tell you, your patient should get drug A, but if you give her drug B, she's going to die tomorrow, but I don't know why. I have no idea why. That's not acceptable. You need systems that can also, just like a human, converse and let you in on what is the knowledge or hypothesis or chain of knowledge and hypotheses that the system is using to make that recommendation, or to make a counterindication and advise you not to take that drug. So you'd like to say, well, I think you should get drug A and not drug B because I've analyzed your genetic mutations in your cancer, and I can see that the tumor is driven by a mutation in AKT. But I also see you have, the following 3 other mutations, which lies downstream of that, and therefore I would predict an AKT inhibitor would not work. However, a JAK inhibitor would. That's the kind of conversation that and rationale behind that conversation that we need to elicit from these AIs. Related to that, I'll say less about transferability, but you of course also need to make sure that, or at least assess the degree to which your model makes accurate predictions and that are backed up by mechanistic rationale for how large is the swath of patients for which those are valid, would be the question of transferability. Is this a system that you have evidence essentially works pan-cancer across, say, all solid tumor types, or is it really most efficacious for late-stage breast cancer? Would be the question of transferability. There's also a question of transferability not between cancer types, but among ethnicities. And so there's an increasing number of illustrative cases these days, both in the literature and in the news, about diagnostics that have been developed in, say, white males that don't work in other ethnic groups. There's other cases where you explicitly know that this set of markers only works for a given genotypes, that is to say, ethnic background. So those are the 3 challenges, and there's undoubtedly more that I'll talk about or that I'll touch on today. Now, what I want to do now is talk about how we get to this mechanistic interpretability. How do we build these AIs, and especially how do we teach it not just to recognize patterns of patient data, but to make full use of knowledge of biology, the cell and tissue biology of cancer, in being able to give you that mechanistic explanation for its actions. So I'll call this biology guided AI. We need these systems not just to recognize patterns in the patient data, but we need them to understand the biology behind those patterns so they can discuss, just like human researchers and physicians discuss a case. So where does that knowledge come from? Well, it doesn't come directly from the genetics themselves. The genetics themselves, you might be aware, are really just reading out the sequence of your human genome. When you get cancer, there's many different kinds of alterations that can be associated with cancer, but most consideration has been given to the direct genetic alterations to that human genome sequence that happen. Typically, you can be born with some alterations called germline variants that can influence your cancer risk or developing cancer later in life. But the other critical thing is for a cancer to develop are so-called somatic mutations. These are mutations that are changes to your human DNA in cells after you're born that happen, as you walk this earth. In fact, we could go on about this for 30 minutes at least, and people do, but there's an increasing body of evidence out there, or data, I should say, charting exactly how fast these mutations do build up as we age, and how much exposure different tissue types have to different kinds of mutational stress. But it's a fact of life. the longer we live, the more mutations that accrue. Many times those mutations are perfectly benign. In fact, in the vast majority of cases, it's thought that those mutations are utterly benign. And places where you get especially large numbers of mutations, most of which are benign, are places of your body that are exposed to mutagens. So UV rays in SoCal are one of the biggest sources of mutations for us. And so, if you sequence my arm here, presumably I don't have cancer there, and you compare that DNA to a place, a sample from where the sun don't shine, you'll see millions and millions of changes in my arm due to UV rays. Of course, UV is not the only way that or the only type of damage that can affect DNA. It's just one that is accessible, I think, to us, and especially in Southern California. But there's, clearly cigarette smoking would be the other classic way if you want to create a huge number of mutations over time in your lungs. But that's just the changes to the genes. So what about the biology that involves all of those and interconnects all of those genes? I'm not the first person to describe it this way. It's often described that the genome is really, and the genes really just like the parts, the nuts and bolts of the components of cells and tissues. You can think about genes. Each gene is a code for a particular protein, and proteins are like the nuts and bolts of this first generation Golf, laid out here on the Volkswagen assembly facility. But genes and their proteins, of course, those parts don't act in isolation. They come together in increasingly complex sub-assemblies and assemblies, all for the different aspects of the complete machine, which in this case is the full Golf. But of course, here we're talking about tumor cells and tissues, the tumor mass, so to speak. So where is that shop manual shown over here at the lower left? And the answer is, for cell biology, we don't yet have such a shop manual. That's still the subject of ongoing research. We do now, for the top half of the screen, have very well worked out ways of sequencing your DNA. As I alluded to already, these sequencing machines are now a part of clinical life, and to a certain degree, your genome sequence can be covered by medical insurance agencies in the cancer context. In fact, if you've ever been down to UTC Mall, just behind UTC Mall is a company called Illumina, which is the world's, I think it's fair to say, I don't know, largest, but certainly most successful manufacturer, probably the largest, of DNA sequencing instruments. And so that's all above the line in terms of getting the raw code. What we're talking about now is what happens below the line. How do we take these sequences and understand what's the structure of the whole cell? So when you talk about structures of cells and what these proteins enable, scientists like me and many of us, of course, love to stare at these types of 3D structural maps of proteins and protein complexes. There's a whole database of these called the Protein Data Bank, or PDB. Here's a very old protein complex structure called the proteasome. This is the machine inside of cells. It's the vacuum cleaner or the garbage collector of cells. It recognizes old cell parts and sucks them in and degrades them. And every color here is essentially a different kind of protein part encoded by a different gene. But here we're looking at the whole at the whole physical machine. Down here below that is a much more recently deposited and worked out structure. This is, as you might imagine, a structure that was published and deposited in the database during COVID. It's the SARS-CoV-2 RNA polymerase for the virus. And you can see here they've even got the RNA itself as templated in here, and then the all the blue and aquamarine and purple colors are again different protein subunits, each encoded by a different gene of SARS-CoV-2, NSP8, NSP7, and so on. So this is the sort of field of structural biology, and the classic mode of doing structural biology is to use instruments like X-ray crystallography and NMR and increasingly cryo-electron microscopy to work out these one structure at a time. The challenge is that there are thousands of such structures inside of a cell, and that's even just talking about one cell type, much less looking at how these structures differ between different kinds of human cells. And to illustrate what's really not known, and to allude to what we really, the lion's share of cell biology we don't know, I put over here on the right a completely fictitious drawing of the neuronal synapse. This is not a real structure, unlike the ones on the left. It's simply an artist's rendition of what she thinks the synapse might look like at the junction between neurons. This thing has, what we know about the synapse is it has about 1,200 distinct protein parts that make it up. Okay, so many more. This is not to scale. The synapse, if the proteasome or if SARS-CoV-2 RNA polymerase were Earth, the synapse is like Jupiter or something. That gives you the idea anyway. It's a very large structure, and some even call it an organelle for that reason. And we're only beginning to understand what each of those proteins does, much less how they form this 3D shape. And again, one worries that's the not the exception, but the rule when it comes to really what we understand about the structure and function of human cells, including tumor cells. So what we need, or what some of us set out to do a couple of years ago, is really ask, how do we turn Moore's law, so to speak, on structural cell biology? That is to say, how do we make this 10x, 100x faster, cheaper, and more systematic to get at what's the architecture of a tumor cell? And to do that, we turn to two measurement techniques in particular for probing cell biology. One is fluorescent microscopy, as is shown up here at the upper left, and the other is mass spectrometry, and in particular mass spectrometry coupled to affinity purification type techniques. So the idea, just at a very high level, the current state of the art in cellular microscopy is to raise antibodies, or to target one protein at a time with an antibody that will light up that protein. So you here that's in green in this image, and that particular protein will now change from experiment to experiment. So in the next well over on your plate, you'll have a different protein you're targeting with a different antibody, and so on and so forth, ultimately walking down all proteins, or many human proteins. What's always the same from experiment to experiment is the other colors here. So there's typically a constant stain for the nucleus. Here that's shown in blue. There is a constant stain for microtubules, here shown in red, and then what's not shown is you often have a cytoplasmic membrane stain as well. So you can think about those as kind of common landmarks that you always light up, but then you want to look at relative to those, what is the position or distribution in the cell of your target protein, and that target changes from experiment or image to image. So that's resting on 400 plus years of microscope technology. What about the bottom pipeline? Well, the bottom pipeline is essentially resting on, it's the leading edge of thousands of years of biochemistry. So biochemists, unlike microscopists, they don't look at things under microscopes in 2 or 3D. They grind them up and pour them in, in blenders, and pour them over so-called columns to see what sticks. That's essentially how it works. And so you still have an antibody against a protein, but now I want to see what else sticks to that protein inside of this gamish. To look at what those contacts, what those sticky other proteins are, I use mass spectrometry to identify those proteins. For this talk, it's technical enough already, but you can feel free to ask me exactly how that works. But for now, it's just you're just trying to identify what are the proteins that stick to your target protein, and using that knowledge from the mass spectrometer, you can draw these network diagrams to show what pairs of proteins appear to be sticking to one another. You lose, of course, the two-dimensional spatial distribution information that you get in microscopy. Now, you can think about these two kinds of experiments also have a bit of a physical scale associated with them. So classically, people have ascribed the scale of microscopy or the resolution of microscopy to be down to about 200 nanometers, 0.2 microns. A human cell, I've upper bounded this, it's just the size of a human cell at about 10 microns. So that's kind of the resolution classically microscopy, at least light microscopy has been associated with. Now at the bottom, of course, we're now looking when we've lost that type of 2D resolution, but what we've gained is very, very low resolution in terms of the ability, or high, but whether you call it low or high depends on context, but able to see and distinguish protein-protein binding relationships where those two proteins might be within 10 nanometers of each other. Now, these are the classic ranges for these two different kinds of technologies, but you can imagine that maybe there is some overlap in terms of what they can see between the two. And that's exactly where my lab picked this problem up a couple of years ago now with collaborators at Stanford and Emma Lundberg's lab, and at Harvard in Steve Gigi and Ed Hudlin's lab for the biochemistry, and Emma is for the imaging, respectively. What the team of us did is we realized that you can in fact pour both kinds of data into a artificially intelligent algorithm, which can recognize proteins that are positioned similarly relative to one another between the two data sets, and based on that reinforcement, start to find groups of proteins that clearly belong together and start to build large maps of those so-called protein complexes. And I show this slide. I don't insist to everyone or expect anyone to understand exactly what I'm showing, other than you're building a map. So each circle here represents a protein complex, and there's hierarchy or there's hierarchical relationships as you navigate from very large compartments, moving up on the page to smaller and smaller parts, just like you would for any human-made machine in terms of having a parts list of a car, for instance. But again, I don't expect, I'll show some examples shortly. But here, just absorb perhaps that these are complicated diagrams that have ultimately hundreds of protein machines that are resolved in these types of integrated data sets. The other thing you can do is as you start to explore these diagrams is when you see protein complexes that are so some of them are well known to cell biologists, and about half of them, it turns out, are not well known to cell biologists as we map tumor cell populations this way. And so when you find a protein community or complex that is not so well known, you might have heard of, or maybe you haven't, AI approaches being applied to get exact structures of these complexes. And we use something called AlphaFold and its variant AlphaFold multimer to do that. And this again has been made possible in just the past couple of years with the release of AlphaFold, which has absolutely transformed the ability of structural biologists to make predictions of structure from protein sequence. Here's an example of the discovery, essentially, or at least the significant expansion of a known complex is really the more accurate way to say it. This is a known complex I'm showing over here, right, called the RAG regulator complex. It's important in cancer, it turns out, because it's involved in the decision point of cells. Do I grow like gangbusters, or do I clean house and garbage collect and tidy up? Cells can either grow like crazy or you do some housekeeping through what's called the mTOR pathway. And it's kind of the gatekeeper of that. Now, there were two two novel proteins that were clearly grouping with this complex in an unexpected way. And those two proteins were ITPA and BORC-S6, shown over here at the left. So here's what the images look like for these for 6 of the proteins that came together here. I'm not showing you, that's the imaging data. What I didn't have for on the slide to fit in is the biochemical data with mass spectrometry, but it's also very supportive of these proteins coming together. And then you know, given we already had a very good structure for the rest of the complex, it was relatively straightforward to add in where ITPA in yellow and BORC-S6 almost certainly are in these structures. Here's an example of a structure that is well known to biology, but now moving to how we can use these maps of cell machines to decode cancer. What was not known in this case is that this component of the cell is heavily mutated and involved in the pathogenesis of multiple cancer types, including melanoma and lung cancer. That component I'm talking about is collagen. Here is what collagen looks like in the upper left here in a textbook, Biology 101 sense. First of all, collagen, is the major component of what's called the extracellular matrix. So the space between cells that really collagen fiber knits all the cells together. Of course, in terms of skin care, you always read about supplemental collagen in a variety of ways. But collagen, of course, is just it's ubiquitous in this extracellular space. You can think about collagen as not a single protein. It's made up of 3 proteins drawn from a very large family of collagens, which come together in a braid. And that's shown here in this textbook picture. You kind of choose 3 of these different collagens, like a collagen 3A1 and a collagen 12 and a collagen 5, and they get put together. And then just like a rope, you then braid those fibers together with others and again to build up this large so-called collagenous fiber. Okay, so that's collagen that binds cells together. And it appeared in our map and that was a good thing. It was sort of a check that, you know, we should be able to find most known components of cell biology. But then when we started using these maps, and this is kind of a segue to the last section of my, or the second half of my talk, when we start to use these maps to diagnose cancer patients, we found that a lot of patients had mutations that hit collagens of different sorts, and it's not a single gene. So previous approaches that just sequence the tumor genomes, they miss this because it's not one collagen where the focus is. It's kind of spread out over the whole complex of collagen. So it was missed. What you can show, and this is shown over here, is what we did with Stephanie Fraley's lab over in bioengineering at UCSD, is if you create some of the mutations we were seeing in both melanoma and lung adenocarcinoma, and you'll see here, I mean, it's the majority of melanoma. 70+% of melanomas we can find a collagen, some mutation somewhere in there. And if you look at where the mutation is embedding, it's typically disrupting this primary braid. It's causing it to be destabilized, essentially. So what Stephanie's lab was able to do is go in and make some of these mutations in a model cell line. She's an expert. She's a world leader in understanding collagen in the extracellular matrix in tumorigenesis. So what she had the know-how and technology to do was to make those mutations in cells that deposit collagen and make that matrix. Okay, so she does that, and so then after those workhorse cells have done their thing, you now have like a flat bed of sort of gelatinous collagen. Okay, and either it's the mutant form, or of course she also makes as a control the normal form. And so you can kind of have two collagen beds, and into those two collagen beds, she now seeds a common cell type. Here, A549 is a type of lung cancer. It's a lung tumor cell. So she comes in with the same exact cell type and then watches the behavior of those cells in one collagen matrix or another collagen matrix. What you can see here is there's a big difference with this marker that she's using. This is called Ki67, and it's a marker of cells that proliferate like crazy. So you can see, for whatever reason, you put those lung cancer cells in this mutant matrix, and they proliferate like gangbusters. And you know, the story to be continued. But definitely that was convincing evidence that these mutations have a functional effect towards tumor pathogenesis. So now let me turn in the last part here, and I'm a little more than halfway through, to talk about now back to the art of the AI systems that we now find ourselves in a position to construct, given that we can now get some kind of a higher level or global, I should say, cell-wide map of what are the protein complexes in a tumor cell using these imaging technologies, the microscopy and the mass spec. Here's where we've had a lot of papers come out recently, which is why this talk is timely. I've listed some of those over here. We're basically going through now different major cancer types and drugs that are standards of care in those cancer types, and building AIs that will use all this information to predict whether a patient is going to likely be responsive or non-responsive to some of these drugs. In fact, I thought I saw Zhao, the first author, is on this call. She just had a paper come out last week or the week before in Cancer Discovery on building an AI that can recommend whether a patient will respond to chemotherapeutics or not. Replicative stress is the main mechanism by which most chemotherapies work. So not all, but most drugs like cisplatin, gemcitabine, olaparib, these are standards of care cancer therapies, and they all work by increasing essentially they're genotoxic agents which increase the replicative stress load on cells. And of course, since cancer cells are proliferating very rapidly, they've got to replicate their DNA every time they divide. So they're very susceptible classically to replicative stress, DNA damaging agents, essentially. So I'll talk a little bit about that, and then we have a companion paper that is also in press on response to a particular targeted therapy that's standard of care now in late stage breast cancer, called a CDK4/6 inhibitor. And drug names that are CDK4/6 inhibitors would be palbociclib or ribociclib would be examples you might or might not have heard of. And then we have, of course, this is part of a larger spectrum of research, both in the future and in the past. But the way to think about it, and again, I don't expect you to understand everything on the right, just to show you a bit under the hood about how these models work. You first provide and train these models on as much data as you can really throw at it on drug responses in 3 different contexts. So of course, eventually or ultimately, you want to directly predict, given patient data, patient mutations, and historical data on which drugs those patients have either responded to or not responded to. And we do have some of that data. However, where most of the current data lie are not directly in patients. We have not hundreds of examples, but thousands and millions of examples of drug responses in the lab, either in rodent models, and those are examples of that would be what's called a patient-derived xenograft, where you take a patient's tumor and you basically you create a cell line from it, and then you plant those cells under under say 20 different mice skin, and now you give each of those mice a different drug to see which one is gonna which drugs make the tumor shrink, which drugs don't make the tumor shrink. Okay, as those tumors grow out in mice, that would be an example of what people do in the thousands of experiments that we have at labs like mine can access, and those are called PDX tumors. Then we're moving to the left even more. You have millions, not thousands, but millions of data points where you're just growing cells in a dish. So there's no live animals involved. You're simply growing a population of tumor cells in a dish and exposing that population to each of thousands of drugs. And then you do that for thousands of tumors to get these millions of data points. So here is kind of where we have data on mutational profiles, molecular profiles of tumor cells matched with what drugs worked for those tumor cells and what drugs didn't work. Okay. Now, is a tumor cell line response as good as a patient tumor response, as good as a mouse? No, of course, these are the highest quality, the best data to have, of course, are the patient data, but you're just limited in how much data we have at present. The risk with tumor cell lines is a tumor cell growing in a dish may or may not behave like that same tumor would have or did when in a patient or even in a mouse. So this is a well-known challenge over all of biomedical research. This long-standing complaint about the difficulty of translation from one model to another, but nonetheless, on the bright side, we have a lot of data in model systems, and if even a shred of that information is relevant to patient tumor responses, you'd like to pick that up and have the model absorb that knowledge, and it turns out that's what happens. So you basically, our current pipeline is we start off in tumor cell lines where we have the most data, we train the most there, and then we either just test directly in patient tumors or PDX tumors in mice, or in some cases we'll go back and do some fine tuning of the model, and so some more training essentially of the model in mice before moving to humans. But you kind of move from left to right on this slide as the models get more and more mature. And then built into the guts, and again, I don't expect you to understand or to lay this all out in detail here in this talk, but then built in the guts of this model is knowledge of these molecular machines that you've been able to detect. This is sort of tumor cell architecture. So let's see an example of this, and I'll use here this example of CDK inhibitors. And the model we have built to predict outcomes, which patients are going to be resistant to CDK inhibitors given their molecular makeup. So we've trained these models in cell lines. That data, I'll just, in the interest of time, skip over it. That's on the upper right. We can show that the models validate pretty well in PDX mice. That's shown on the upper or the lower right. But let me focus on the patient data. So in the center left, and most of the slide is showing a retrospective clinical trial we've been able to perform using this model on, and the patients come from this repository called Project Genie, where again, this is not forward-looking, it's looking historically at patients who have received CDK4/6 inhibitors or not. Left got the inhibitor, gray, right, does not get the inhibitor. Everybody gets endocrine therapy as standard of care for this group of patients. And now what's shown in the rows is what our AI model said should happen, having looked at the mutations that these tumors, each of these tumors have. And so based on, and it turns out it's looking, as we'll see here in a minute, it looks at about 90 different genes is what it's learned to look at for deciding whether mutate, and using mutations in those genes to decide whether or not the drug is going to work. So based on that, it does a computation. It's very fast once the model is trained. The training is slow, but once you have the model and it's clinically deployed, or at least in this research setting deployed, it's essentially instantaneous to get a prediction. We ask the model to kind of bin patients as sensitive, partially resistant, and fully resistant. Okay, and you can see the numbers of patients that got the CDK4/6 inhibitor that the model thought should respond. 34 of them said, oh, these should respond. 16 will sort of respond, and 17 won't respond at all. Over down here at the lower left is the so-called survival curve, or sometimes called the Cox a model, where is shown the results. So each time is on the x-axis, and the percent of patients still surviving is shown here on the y-axis, and you can see that patients did pretty well over time when the model said they should do well, and pretty poorly when the model said they should do poorly. And for reference, the gray curve here is the effect of not receiving CDK4/6 inhibitors at all. So essentially, the strongly resistant patients behave similarly to patients who simply did not get the therapy. Now, like I said, this is not the advance here is, well, so first of all, there's we think this result is fairly unprecedented, just in accuracy. So if you think back to the beginning of my talk, I'm not going to go through those bullet points. So this is it perfect? No, but it's exciting, and it's a significant improvement in accuracy for what people can predict now. To make those predictions, though, it's not looking at just one gene, one mutation or one gene. It's looking at a constellation of mutations across 90 genes. This is what the models learned, okay? And those 90 genes, it turns out, make the subunits to 8 of these different molecular machines. And again, this is what the sort of internal guts of that look like. I'm not going to take make a quiz later for what this what all this represents. But there's of course a complicated wiring diagram inside of the guts of the model that guide it for how to do this. And what you see among these 90 genes that make up that encode these 8 complexes is you see some complexes that are well published, people already know about as important for determining whether these drugs are going to work. And then you also see some complexes that are complete surprises. And here up in the upper right is an example of a complex that comes out that's used by the model. It's very informative. If you look at this set of mutations in these genes, it's very informative for predicting resistance, it turns out, to CDK drugs. And in fact, we found about 25% of resistant patients have an amplification, meaning the gene has been dialed up in genes by the name of TBL1, XR1, KAT6A, and run X1. What we can now then do is, just like I went in and did an experiment to prove those, some hypothesis, I can now go into an in vitro model, a dish, and dial up each of those genes, and look at the effect of doing that on the drug response. And so the increased in the purple bar here compared to the gray bar shows an increased drug resistance that we observe when we do that. So I think oh right, and then kind of end. I have one more slide just to kind of end on a fun note here. The other aspect of AI I can touch on my talk. It's a very different one. A lot is we've been very impacted, I would say, by the ChatGPT type bots that people here on this call. I'm hoping some of you have already used. I've certainly since GPT-4, OpenAI's product. I just say GPT-3 first came out, over a year ago. I think I was at a dinner party in 2023 around the holidays, or 2022 around the December holidays, when another professor said, "Hey Trey, you've seen GPT? It's fantastic," and I just couldn't believe it. And so then, there's many of us who spent 2023 exploring the uses of these things in science. So the use that we've really, I think, been amazed by here is the ability to take, there are so many cases in molecular biology and genetics where you have a set of proteins or genes you've just discovered in some experiment, and you want to know what those genes do. Here in our case, it's, here's this last slide. Here is a set of proteins that mutations, amplification events, genetic alterations cause drug resistance. So what is this thing doing? Well, let's ask GPT. That's the simple thought. So this is shown for not this set of genes in the last slide, but just a different set as a sample. If we feed it a group of genes that maybe were detected, elsewhere in this map, TBX5, ISL1, TBX3, and so on. Maybe these are forming a complex. And we ask GPT, hey, GPT, imagine. So first you have to give it some context. This is called prompt engineering in the lingo. You say, hey, imagine, can you please role play the role of a scientist or an assistant to a scientist. Imagine you were an assistant to a scientist, okay, a biological researcher, in fact. I'm going to give you a set of human genes. We have good reason to believe these genes work together. okay, in causing drug resistance. What do they do? What is that function they work together on? Do you know? And the reason why it might have an ability to know is because these bots, unlike mere mortals, human scientists, have actually read all literature in the public domain. So if it's if it's a paid journal like New England Journal of Medicine or Science, maybe it hasn't seen it. But for any papers that are posted publicly, and there's an increasing number of those in biomedical science due to the existence of something called PubMed Central, where a lot of people post, most papers. This thing is read, arguably, the vast majority of the literature. Okay? And it's the only entity that's read. There's no human entity that's come close. So perhaps it's in a good position to tell you something insightful about the proteins that come up in your experiment. And so you also so you ask it a couple things. You say, okay, so you kind of set the stage with your role play, and then you say, so now what I want you to do, GPT, is come up with a short name of what that function might be that these genes do based on your extensive reading and knowledge. And then can you please write 1,000-word, back to the interpretation explainability, we want you to rationalize that function or that recommendation with a 1,000-word essay, please. And that's essentially what you type in. And here's the result. So it says, I think these genes function in cardiac development and regulation. And the reason and we should call this function cardiac development and regulation. And here's why, the first 5 are transcription factors and signaling molecules with critical roles, ... And then, it goes on for 1,000 words and says, to summarize, the proteins in this system are involved in the complex process of heart development and regulation, ... Okay. You also, by the way, can get it to reference, to drop references, and you can have another bot come in and check those references. That's not shown here. The other thing you can do is it turns out that just in the past month, we have seen the public release of other fairly powerful large language model bots. GPT-4 is still probably is still the leaderboard winner, but Google just released Gemini early last month in 2024, and there's a French AI company which has released now Mistral and Mixtral, which is, unlike GPT-4, is an open source, I believe, effort. So it's exciting in that maybe all of this will now be in the public, or this ability will be in the public domain. Notice that actually Gemini and GPT-4 agree largely on what these genes are doing. Mixtral doesn't, so maybe that shows the gap between the open source model and the paywall models. But on the other hand, maybe it's found something, it has some insight the others don't. So by the way, the next step that again is happening in mainstream computer science and is going to impact our research as well before too many more months, you will get these 3 bots in their own virtual room to hash it out, just like you might have 3 reviewers on a panel, and they'd have to get in a room and come up with a consensus report on something. This is the same thing is happening with these co-pilot so-called experts. So anyway, I've taken you through a tour of AI for precision oncology with some detours along the way and some side stories. But just as a concluding overview, we've gone through the use of microscopy and mass spectrometry to build these maps of the inner workings of tumor cells in terms of all of their molecular machines. We then use these maps together with patterns of mutations and linked to drug responses, both in patients, mice, and tumor cells, to build these deep learning AIs that can look at a patient's tumor and, based on the mutations it sees or does not see there, make a recommendation for whether a drug is going to work. I think at that point, I will simply acknowledge a large team of folks. I think I've mentioned a few of them already who made this work possible. I mentioned Zhao Yu, and I'd like to also acknowledge Akshat and Sung Jun on this recent modeling work. I'd like to acknowledge Yue Chen on building out these maps of tumor cell biology and the inner workings of tumor cells. And Clara Hu and Dexter Pratt on this recent use of the GPT-4 model to answer questions about genes. Thanks very much.